My name is Justin Wagner. I'm the Beef Systems Extension Specialist out in Garden City, Kansas. And it's my pleasure this afternoon to introduce our first speaker in this afternoon's breakout session, and that's Dr. Dan Moser. And he is an associate professor of beef cattle genetics here in our department at K-State. And the title of his presentation this afternoon is DNA Marker Assisted Selection.
Well, thank you, Justin, and let me add to several people have commented. Justin's one of our newest hires, and particularly those of you in the southwest part of the state that if you haven't met Justin yet, that he's going to be a real asset to our team and the good work that he's starting to do already in that part of the state. So we really look forward to having you on our team and sure want to welcome you to our program. An exciting topic for me to address, I think now is the time. To me, there's more happening in beef cattle genetics than there's happened in a long time. Some new tools that are coming online and becoming available. And so I'm going to go through today and talk about some of the things that are available now in the marketplace and maybe some things that may be coming down the road a little later. Some things I want to talk about: marker-assisted selection. Lots of different people maybe have different levels of comfort and experience. I know people in the audience that have already made some investments in this sort of technology and are using that in merchandising animals or in making selections. And there's some people that are probably very new to this whole technology, and my goal is certainly to reach everybody. If there's questions you have as we go through, please do stop and ask me, and if I can clarify something, if I hit you with some jargon that you're not sure of yet or that sort of thing, please do, because my goal is to work with everybody and make sure your questions are answered. But we'll talk about what it is, what it can do, how well it works, and some other things that are certainly, I think, coming down the line that could make things even more exciting, I think, in terms of this area. I'll define marker-assisted selection as the idea of using DNA tests. There are other selection tools, some blood assays and that sort of thing that are also being used in a few places, but what I'm going to talk about today is DNA markers, genetic markers, broadly to gain information about an animal. And there's all kinds of information that we might be gaining. I tend to think of it, because of my interest in genetics, as identifying parents and identifying individuals that have the ability to sire in a natural service setting or an artificial insemination setting superior animals. But there's also a component of this, I think, in terms of identifying potentially superior market animals. And we'll talk about that a little bit too. Really what we're getting after is the battle we've always fought and will always fight to some extent in cattle breeding, and horse breeding is even more this way, that we're a race against time trying to gather as much information as we can as quickly as we can. But compared to some of our competitors, we're really behind just in the biology and the generation interval. We can't turn generations like poultry can. We can't turn generations like swine can. We don't learn as much about animals as quickly as they do, and that's somewhat of a competitive disadvantage to us. And so this technology at least offers the opportunity, perhaps, to learn more about animals much quicker in their lives, so we can make better decisions, identify animals sooner, and make progress from that. I will just take a minute to talk about this, even though this really isn't the subject of my talk, but it's something that I think we'll see more out there, particularly as costs come down. And that's the idea of marker-assisted feeder cattle selection and management. There seems to be some interest in this area and some people working in this area and some application out there in the industry to try to do a better job of value discovery of feeder cattle. And quite honestly, in my opinion, we don't do as good a job of that as we probably should in our industry. We've come a long way since I sat in those seats as an animal science student in terms of discovering value in fed cattle, but the feeder cattle part is a little more elusive for a lot of good reasons. But there might be an opportunity there with some DNA profiling and those sort of things to do a better job of assigning value to feeder cattle. We have made some progress there in terms of communicating data up and down the supply chain and those sort of things, but here's another opportunity that may someday be important. It works quite well. Some of the tools that are out there in this area really, partially of what they're identifying though is breed differences. That these technologies, some of the markers that they are identifying and tying those to a particular trait might be markers that are, or DNA sequences that are found predominantly in one breed or another. And we have to be a little careful about taking technology developed with this thing in mind and trying to take it back to a seed stock, purebred type cattle breeding enterprise and think it's going to do the same thing, because we may be finding what identifies superior feeder cattle for marbling as just genes that most Angus cattle have, perhaps. And those particular, not necessarily genes, but DNA markers that they have, and we've identified them as having more Angus, but that necessarily won't help us trying to breed Angus cattle with more marbling, for example. So there's some issues there in terms of which way some of these are going, and not every test that does this well will help us breed better breeding cattle. Cost right now is the big issue, but like anything, you know, some of you, the younger people, remember when DVD players were $500. Some of you remember when VCRs were $500. Some of you don't remember VCRs, but nonetheless, I always have a challenge in my classroom and start talking about some of those things, and it's easy for college professors to get out of touch with that sort of thing. But nonetheless, the idea is everything over time will become more affordable as the technology becomes simplified and because when the volume goes up. So this may be something that we don't do now, but that doesn't mean we won't be doing it in maybe just a few years or maybe a few more. Another application of DNA technology that is not maybe as exciting, but one that's, I think, very useful in a lot of situations is parentage identification. The same sort of technology that we use in human paternity or in forensics, those sort of things, can be applied and is being applied in commercial cattle settings, for example, particularly when we have multiple sire mating. And, of course, that's a system that works very well in a lot of ranch setups. And so it gives producers an idea to know a lot more about which bulls are doing what. It tells you a few things when you can then take a DNA sample on a calf and match that up to which of maybe the 5 bulls that you turned out is actually the sire. You'll find out some things about bull dominance and about which bulls are doing most of the work. It's not uncommon if someone's turning out 5 bulls on 100 cows or something like that to have one bull sire 40 or more % of the calves, and one bull might not sire hardly any. And so that can give you some management strategies in terms of what is the optimum number of bulls to turn out in that sort of a situation. So there's some good information there. And then about evaluating those sires in terms of which ones are giving you the traits that you desire, the ones that are the highest performing or the lowest performing, which are truly giving you the best daughters, things that a single sire producer would know that you don't know for sure. These sort of opportunities can be done with DNA parentage identification. And of course, DNA can be a powerful source verification tool too. And so that's another whole area that we could talk about perhaps, but just want to throw that out as something to consider as we move more in that direction of identifying source of cattle and those sort of things, that the DNA will always be with them. In fact, we can extract the DNA from the supermarket in some cases, and fresh meat products and trace in that sort of way. Now, talking about changing or selecting for traits of beef cattle, getting more in the seed stock, either as a seed stock producer or a customer of a seed stock producer that's buying bulls and that sort of thing. First, I'll talk about qualitative traits. And qualitative traits are ones that are categorical. They're yes, no, they're categories. They're things like color or horn status or muscle status, double muscle. Those of you familiar with some of those breeds. Presence or absence of genetic defects, yes, no kinds of things. And these are a place that we've made a lot of progress in identifying with DNA tests. These things are usually determined by just one or maybe just a few genes, and that distinguishes them from some of the other more complex traits like growth or marbling. Just a few genes, sometimes just one set of genes that determine these traits, and usually they only affect one trait. That if we change the genes for coat color, that's not likely by itself to change anything else about the animal. That won't necessarily be the truth in some other categories. Some of you have used a DNA test for coat color, and this is an example of one that's currently available. Actually first discovered in mice, the coat color locus in mice, and then transferred we have what are called comparative maps that tell us which part of the mouse chromosome lines up with which part of the cow. And some sharp people at Clay Center pounced on that mouse research and tied it back to the cow map, and were able to confirm what we suspected, that region was pretty similar. And so the result that you'll get, the animals that will tell you which ones are homozygous black and will always transmit black on, which ones are black animals or carriers of the red gene, which ones are homozygous red, and then a gene that some of us didn't even really know existed, but something called wild type, which wild type is sort of an intermediate gene that is sort of neutral, and it allows other things to be expressed. If they have another red gene, then that will result in a red animal, but some of the cattle that show brindles and some of those sort of things would also have wild type genes. As we do some of this genetic testing, we find out this wild type gene is around more than perhaps we thought. In some of the red populations in particular, cattle we assume to be homozygous red carry a wild type gene. And that won't really be an issue in terms of the color of the cattle unless they're crossed with some of the Brahman derivatives or some of the Spanish cattle, the Corriente Longhorn type cattle. But in those cases, wild type can allow some of those other wilder color patterns to come up that you may not have expected. Another one that I'll talk about, and I talk about this not because there's a tremendous interest in double muscling, but it illustrates some things that I think we need to keep in mind as we think about genetic tests and what they do or don't tell us. There is a definitive test for double muscling. It is a single gene found on the chromosome number 2 of the cow. It's a recessive condition, so there are animals that are not double muscled but carry the gene for it, and the animals that are double muscled have 2 copies of that particular gene. There is somewhat of a leanness advantage for the carrier animals that's been identified through work at the Meat Animal Research Center. But what's interesting to me is that the 2 primary breeds that show this condition, Piedmontese and Belgian Blue, have different genes that have the same effect. Okay? I'll show you that with an illustration. And as we think about this I think has some ramifications for how we evaluate tests for marbling and some of those sort of things. To kind of orient you to the slide here, that at the top, this is an animal that is half Piedmontese and half of a traditional breed. Okay? The top gene is the normal gene, that has a G in this position. The Piedmontese allele is just a simple substitution that puts an A in this position. One single DNA base basically makes this gene, which is called myostatin, non-functional. And so this animal would be a carrier, but if they had 2 genes, both with A's there, then they'd be a double-muscled Piedmontese. And that was the breed that the test was first developed in. The geneticists then at Clay Center decided they had some Belgian Blue cattle, and they ran the same test, and it told them that all the Belgian Blues were normal, that they weren't double-muscled. But obviously you could see that they are. So with some further work, they found there was a different mutation in the same gene. Downstream a little bit, the Belgian Blue cattle have the G here. They test normal by this test, but they have 15 bases just gone, a big deletion down in here. Both of those mutations make non-functional myostatin. Both mutations result in double-muscled cattle. But had this one not been discovered, then we would conclude the Belgian Blues are normal. Now you can see the Belgian Blues are not normal. But some of the more subtle things, like a slight increase in marbling, you can't always pick up on. And so we always have to keep in mind that just because an animal, we have a few tests that might represent something favorable in terms of marbling, that's not an assurance until we have every gene identified, and we're a long way from that to thinking there might be something down here that's interfering with that. And we can't expect a diagnostic for a quantitative trait to be 100% sort of a thing. Horn-polled is an interesting one, and one that's been out for a while. It came out as a DNA test on the market, sort of went away for a while, and has come back again. At least 2 companies now have a horn-polled test. But it's not been quite as definitive as the red-black. The red-black, we feel very comfortable that we have exactly the mutation that causes the difference. And I've not, other than an occasional, very rare lab error, there's no case of an animal that had a DNA test for red-black that didn't match up what they really were. Horn-polled isn't quite that way. Remember, polled is dominant. The absence of horns is dominant. Heterozygous animals will not have horns. They may show scurs. Some of them do, not all of them. But some of them, they carry the horn gene and could pass it on to their offspring. Horned animals are homozygous horned. They have 2 copies of the horn gene. And there are, again, a couple of tests, different companies. Igenity, I believe, has a test, as well as MMI Genomics has a test for this. But as from my experience working with breeders that have sent these tests in, that they get a significant number, maybe as high as 20% of their animals come back as inconclusive. That there's probably more than one mutation in the horn gene that has some impact there, maybe a little bit like what we're seeing with Piedmontese and Belgian Blue. But we don't have a 100% accurate test for horn-polled at this particular time. And there have been some that are just frankly flat wrong, that I know of at least 1 or 2 breeders that have had animals classified as homozygous polled, they bred them to horn bulls and got horn calves. And that's a definitive that they absolutely must be a carrier. So I have some level of confidence in these tests, but I think they're still, to some extent, a little bit of a work in progress. They'll get better over time as more data and more work is done. Genetic defects is an area that this technology has in the last 2 or 3 years had a tremendous impact. We've had genetic defects in most of all breeds for a long time. And I talked to breeders that went through the dwarfism issues many, many years ago, and the only solution to dwarfism was to cull any animal with a suspect pedigree. With DNA tests, we can do much better than that. Now that a lot of the genes, the actual mutations that cause these have been identified, that we can now identify which animals truly are carriers and which ones are not. That, again, every breed that I can think of has at least one of these sort of things that's testable. It shouldn't really be an issue for commercial producers because hopefully your seed stock producers are doing a good job eliminating from the population. But those of you that are in the seed stock business, if have questions about these, I'd be glad to talk with you about individual things with your particular breed. But they're all out there and more are being tested. The thing I really like about these sort of tests is that it will tell you which ones are the carriers, but it'll also tell you which progeny of carrier sires are clean. And there are some prominent sires in a number of breeds that do a lot of good things, but they carry a genetic defect. Half their progeny will be just fine. And if we can identify that half and go on with those, we can keep the good things that that particular sire offers without having to worry about the genetic defect. If they test clean, regardless of what their pedigree says, then they are, and we can go on and not worry about genetic defects. Quantitative traits are what we're most interested in because those are the ones that have the most dollar value assigned to them. Things like any weight and measure, whether it's birth or yearling, all our performance traits, all our carcass traits will be categorized as quantitative traits. They're influenced by many genes. They're not just 2 or 3 genes that impact these. There's a lot more than 2 or 3 marbling genes. There may be 40 marbling genes. We don't really know. If there were only 2, then cattle would fall into 3 or 4 distinct categories. They'd either be low, middle, or high. There wouldn't be this continuous distribution of marbling the way there is. So we know there's lots of genes involved. And each gene is having a very small effect. If there were big major genes that were driving some of these traits, again, we'd have them in stair steps, not in the nice smooth bell curve that we see. Just keep in mind that sometimes we see tests presented to us as this is a marbling test, or this is a growth test, or this is a milk test. But keep in mind what we're testing for in the end is genes for proteins that make things like hormones and enzymes and growth factors, differences in the biological process, really not that different than the biological process of other mammals, even humans. And so when we change the level of one of those hormones or one of those enzymes, it might do more than just one thing. And it's perhaps a little naive to say this is a marbling gene and this will change marbling and make the assumption that it won't change anything else. That as we change, just as we change through selection, traditional selection, we change more than one trait at a time because of relationships, that gene selection will do the same sort of thing. So every gene or every test will likely have will have one effect, but it may likely have more than one effect. And some of those things, as you pursue a high level of something that you want, you may also be selecting for an unfavorable part of something else. That'll take more time to have those more clearly defined. We spent a lot of time in animal breeding research defining genetic relationships or correlations between traits, but defining the relationships between genes and gene tests will take some time as well. I'll talk a little bit specifically about a few tests, and the reason I've picked the ones that I'll talk about is they're, in some cases, the one that's been on the market the longest, and they're the ones we know the most about. There are new things coming on the market all the time, and so I can't really cover everything. But I'm going to talk specifically about a couple. One is a marbling product sold by Bovigen Solutions, or you may know as the GeneStar company, but Bovigen Solutions is the American company. They sell a panel of markers. A panel means a group of markers, or a group of tests. They're testing with one sample now. You don't have to take 4 tubes of blood. But with one sample, they will test at 4 locations and use that as a predictor of quality grade. Okay? And the first test they have, which has been around since 2000, is for a mutation near a gene called thyroglobulin. It's not in the gene, it's upstream from the gene, but they've claimed an association with marbling with that. Since then, they've developed others. One of the challenges to someone on the academic side is thyroglobulin, the work was published in refereed journals. It was patented first and then published, so I can dig into the journals and I can read quite a bit about it. The problem with that is that competitors can as well, and this is in a competitive business. That even though they're patented, patents work really well in some countries, but there are some countries that don't respect patents, and for that reason now most all new technology in this area is protected as a trade secret. And so I can't go on their other tools or in the competitors' tools and learn nearly as much about. They claim QG 2, 3, and 4. but I honestly have no idea what they're testing or where they're testing. I can dig into some patents and find some information. I have some suspicions, but I can't say for sure what they're testing for. That's just the way this business goes, and it's really not that much different than the seed corn business or any other sort of competitive business. But there are favorable and unfavorable sequences at those. So the results that you used to get were in favorable alleles or in stars, and so an animal could be unfavorable. They have 2 alleles or 2 possibilities at each of the 4 spots, one from the sire, one from the dam that they have. They'll pass on a random one of those 2 every time to their calf. So there are actually 8 alleles. They could be homozygous favorable at all 4. That would give them 8 good ones. They could be homozygous unfavorable at all 4. That would give them 8 bad ones. They could be heterozygous at all of them and have 4 good and 4 bad. There's lots of different possibilities. This is some independent work that was done through a cooperation of what's called the National Beef Cattle Evaluation Consortium. Their website's on there, and if you want to dig into more information, that it's all out there for you. Consortium's a group of universities, and K-State's been a partner in that's been doing some of this independent validation work. What I want you to get out of this slide is that what they're testing here is the significance. This is a population of commercial Charolais Angus cattle that were part of the NCBA Carcass Merit Project, and they're testing for significance. These are p-values, significance values. Lower numbers are better. You want generally the kind of the standard is 0.05 or lower. This sort of tells you what's the chance of getting that just by dumb luck if nothing was there. Okay? And so the way they've analyzed this is they've said, okay, what is quality grade 1 marker, thyroglobulin, doing? Is it having an effect? Is the second one, is the third one, is the fourth one? None of them by themselves are significant for marbling score. None of them are, one is close to significant, but not significant for percent choice. As a group, though, the number of stars across the 4 does get to significance, so we can conclude that there is some information there about marbling. But particularly 2 and 3, their utility is very marginal. There's not a lot that they really tell you. You know, I'd be much more excited to have bulls that are favorable for the 4th marker, because that seems to be having a much bigger effect than is 2 and 3 in particular. 2 and 3, those high levels say that they're not really showing us much impact. And there's some work out of Clay Center that shows the 1 marker, the thyroglobulin marker, does a great job in Wagyu populations and not a very good job in any other breed. That's a peer-reviewed paper out in the Journal of Animal Science right now. This is how Bovagen reports their results, and as we get more and more, we went from 1 set to 2 to 4, the number that actually tells you better than the number of stars or the number of favorable results is what they're calling a GPD. That's telling you something about the effect, okay? So for example, if you had an animal that tested favorable, 2 favorable at all 4 locations, they're predicting that animal itself, not their offspring, but that animal would have a 33% increased likelihood of grading choice. And that goes down to 0. They set their scale so there's no negative numbers. But the most unfavorable animal down here that's unfavorable at all of them. So you can see this in differences of percent choice of that animal. Okay? It looks kind of like an EPD, but it's not. It's much better than saying they're 4 out of 8 or 6 out of 8, because as I told you before, some stars are definitely bigger than others. Some are having more effect. Some genes are bigger than others. And I think this is the way that we'll move into reporting these results. But what does that number really tell you? It tells you about that animal's potential, not their progeny's potential. Okay? So to get it to more like what you're used to with an EPD, the first thing you have to do is divide it by 2, because an animal passes on half their genetic merit. It also includes some interactions and other things that aren't passed on to offspring. And so if I look at those numbers, I'm going to at least divide by 2, maybe divide by 3 or 4 might be a better description of how much difference you would actually see as you compare those. The other thing to remember, too, that it looks like a big difference between the very best one and the worst one, but there are very, very few animals of any that test favorable at all 8 or all 4. I don't know of hardly any. Most animals will be a mixture of both, and so that looks like a pretty good range, but other than some Wagyu animals, you won't have a whole lot that come up favorable across the board. If you do have one, call me, but I don't think there are very many. Gene frequencies are also somewhat of interest, because you might be asking, what are my chances of getting a certain result? For example, this shows, this is the QG1 or the thyroglobulin gene. When they tested a population of Angus cattle, and they're not necessarily saying that all Angus cattle are like that, but when Cornell and others worked that, that 22% of the genes were favorable and 78% were unfavorable. Okay? And as you go across there for like 4, all these, there's different variations. Very few animals had the favorable number 3. So it's, you shouldn't expect that. But in some breeds, there's very little variation. Breeds share relationships. They share genes to some extent. And so you might be working with a breed where all the animals are 0 at one sort of thing. And so if you have one that has the desired one, that's certainly advantageous. The other way of thinking about it is there are some breeds where they're all good. And you're not learning anything you don't already know. The other test I'll talk about in detail is the tenderness test. That there's a set of tests that partially developed in the U.S. at Clay Center, partially developed in Australia and Canada, that test for 3 primary genes that impact tenderness. And this is really the most powerful use of this technology to a point because it explains a lot more variation. Calpain and calpastatin, which the meat scientist, Dr. Unruh and Dr. Schaefer could tell you a lot more about, but those enzymes affect tenderness post-mortem. After the animal's harvested, they go to work and tendernize meat. The aging process that you observe in meat is partially the result of those enzymes. Now, if you think back to the other chart, and these numbers were hovering around somewhere to 0.5 to 0.1, and in some cases lower than that. This is 1.8 with 4 zeros in front of it, okay? And this is 4.7 with 6 zeros in front of it and a decimal point. These are very, very highly significant, and these have tremendous power to explain differences in tenderness. If that's one of your selection objectives, you can make tremendous progress in changing tenderness, again, assuming you have variation in your herd. It may be, though, that all your herd has the same profile across the board, and so there's not much opportunity there. But if you're buying bulls and want to change tenderness, that can be done. This shows the magnitude of the effect. Again, these are not EPDs, they're the differences in the animals, but they move as far as from being animals that are all good, the C's are good. 2 pounds of shear force is a pretty big difference. It's something that you would notice, sitting down with a knife and fork. And again, the gene frequencies somewhat depend on breed. For example, the CC favorable is very rare in Brahmins. And we've documented plenty of times, and lots of places have documented the tenderness issues that appear in Brahmin cattle. This unfavorable one only found 1% of Brahmin genes, where some of the other British breeds and continental breeds show a mixture of those two. So they can take some advantage of this, but there's not going to be a lot of animals in that breed that move forward. There are other traits that are currently available in the marketplace. Bovagen has released a feed efficiency panel. That has not yet been independently validated by universities that I'm aware of. We are collecting some feed efficiency data here. Lots of other universities are, and I suspect in the near future we can tell you in a similar sort of manner how well those things work, but we're not to the point yet where we can document those. The Igenity group has a number of traits that they are profiling or assigning numbers to. Their numbers are a little different. Rather than putting in the units, they just give it a 0 to or 1 to 10 scale. That's a little harder for me to get my mind around because 1 to 10 could be, it depends on how big an effect those genes are having. We don't know how much better the 10 is versus a 1 is in the units of the trait. But it is useful for ranking animals. And they have a number of traits that they're profiling, but again, we don't have yet any independent validation of those, and so it's hard to know what we really get for those. The next big thing, and something that we'll see taking off now, is what's called whole genome selection. If you thought this couldn't get complicated enough already, that it will get more, because we have new tools in the lab that will let us run lots and lots of tests very rapidly and very inexpensively. And so we might be testing an animal at 1,000 different places over all 30 chromosomes, and then tie that into an EPD-like analysis, a complex analysis for sure, but all those results where we may come up with using all those markers or a portion of those markers, that work is underway in a number of places right now, and we're going to be a partner with a couple of places, with Clay Center and with the University of Illinois, on a couple of those projects. To wrap it up with, in terms of using these tests, the number of tests for some people is already overwhelming, and it's not going away. And so I think that makes it hard. My opinion, and of course I have an EPD sort of background in quantitative genetics, is to me the most user-friendly way to do it is to incorporate it into an EPD calculation. That work is underway as well. Simmental has done a study where they've incorporated the tenderness markers into their tenderness EPDs. It works really well. They've looked at the marbling markers and concluded that the information there doesn't really change the EPDs very much, that the pedigree, the ultrasound data is still considerably more powerful than what they were able to glean from the marker results. I think that in terms of using these, if you're looking at young animals or looking at a group of half-sibs, for example, that you've got ultrasound information to make selection, you've got EPDs from pedigree in that animal, and those are certainly the most powerful tools at this point. But if you're choosing among full sibs that these sort of profiles, they will vary somewhat. You're trying to figure out which one got the best draw of their siblings, and I think that's where this can be used. But I caution you that if you're looking at AI sires that we have truly highly proven, high-accuracy EPDs with lots of progeny data, that this doesn't really tell you very much new information. That I wouldn't jump off of a bull or decide not to use a bull with great EPDs because his markers don't look very good. Because the EPDs are reflecting all the genes, and the DNA tests are reflecting the discovered genes, which are certainly, I think, in a minority at this point. If you want more information, there's a couple of websites, and if you don't want to write it down, you can come see me afterward. The consortium has all their validation studies available online. UC Davis has really taken a lead in this. They have an extension biotechnologist. That's kind of an interesting position, but a professor named Alison von Enum is doing a great job of disseminating this information, and that's her job, is to relay this information to the livestock industry, and that's a good place to look around too. With that, Justin, I'll break it off. I think it's time for John's presentation, but I'll stick around and we can take questions afterward. Okay, that's good. Thanks for your attendance at Cattleman's Day, and thanks for your attention. It's really an honor for me to visit with you folks.
Next speaker on this session this afternoon is going to be Dr. John Yeager. John is the beef cattle scientist at the K-State Ag Research Center in Hayes. And with that, I'll turn the floor over to John.
Thank you, Justin. I'm going to regress from maybe. I'm going to regress from bull fertility for just a minute and kind of give you my perception of producers' mentality of cows versus bulls, and have to admit that this includes my mentality. And cows have really been revered through the centuries and actually worshipped, and rightly so, because they're either lactating or gestating or both, and most importantly, they're producing a calf, which is our livelihood. We really rely on these cows to produce calves. Cows, being females, are multitaskers, something I'm not very good at, but they're either harvesting forage, producing milk, caring for the calf, or getting rebred. And during the 6 or 7 months that that calf is at the cow's side, she's also pregnant with her next offspring. So she's a really good multitasker. She's given us a calf at weaning time that we can sell or feed out for beef. And so it's pretty easy to get attached to our cows, and we get really fond of them. We're proud of the calves that they're producing. On the other hand, don't get me wrong, bulls have been worshipped for centuries, probably more so by purebred producers than they are commercial producers. But we do our homework. We try to pick out bulls that are going to best fit our production scenario and our operation, and we go to the sale and we get him bought and we get him home, and we're really proud of that bull, and we're excited about the potential genetics he's going to infuse into our herd. And we're expecting him to work for 60 days. That's all we want out of him. 2 months out of the year, whereas that cow's working 12 months. And we're excited about the number of calves he's going to produce, and we get him turned out for the breeding season, and it looks like cows think he's okay, and he likes the cows okay, and we think we're going to get quite a few calves from him. And we go back to check, and there is a cow in heat, and there's a bull with her. And then we see 5 or 6 other cows in heat, and we can't find any other bulls. And we start hunting, and we find 1 in the shade, 2 fighting. We keep hunting and hunting, find the other one on the wrong side of the fence and the neighbors. And breeding season starts to feel like a year long instead of 8 weeks long. And by the time we get to the end of the breeding season, we're pretty sick of bringing this bull home, and we can't wait to pull him on and stick him someplace we can just forget about him. So they got a 10-month vacation, and what are they going to do? They stand around and they eat, and they eat, and they eat. And during that time, we just hope they don't tear up too much stuff. We hope they don't fight too much and hurt each other. And we hope the neighbor's not mad at us because we're bringing them home all the time. But what else is happening during this period from the time we pull them out from the cows? They go through the year, the quality of the forage they're consuming is declining through the summer, into the fall, through the winter, and we tend to pretty much forget about the bulls. We want to forget about them because they're causing us so much trouble. And we really need to pay more attention to the bull's nutrition, because development of the sperm cell requires 60 days. And so whatever occurs to that bull during that 60-day period is going to affect the quality of the semen that he produces. So if we get a late spring snow and those bulls are laying on frozen ground, we could have a detrimental effect on the quality of the sperm that they're producing. And we have to think about their nutrition, provide them adequate nutrition and supplementation. Now, Dan mentioned generation interval, and it's really a disadvantage to us in the cow-calf industry. The generation interval for beef cattle is 4.5 to 6 years. You've heard a lot today about what happened to our industry in the last century or even longer, but think about how much has changed in the last 6 years. And really, the genetics in these cows that are 6 years old are getting outdated considering what's going on in our industry today. So to me, that just highlights the importance of ensuring the fertility of our bulls so we can infuse the most current genetics possible into our cow herd. We've all seen mating ratios of bulls. As the bull's age increases, the number of cows that they're likely able to service increases. And you have to remember, we're going to decrease that ratio with large pastures or rough range conditions. And right now, in terms of the economy and feed costs, we're probably trying to look any place we can to cut corners. And be nice to buy 1 less bull this spring than you typically would. But you have to think about their siring calves, and that's our income. So we got to make sure that we maintain a proper bull-to-cow ratio. And that's demonstrated a little bit with this slide. If we look at a bull-to-cow ratio of 1 to 20 up to 1 to 44, we're still detecting about 100% of the cows that are in estrus. We go to 1 to 60, they're only getting about 66%, and at 1 to 100, they're only finding about half the cows that are actually in estrus. So we have to maintain that bull-to-cow ratio so we can find the ones that are in heat and hope that he gets them bred. I think everybody's pretty good about having their bulls semen checked each year, and when you take them in for their semen check, the vet's actually doing a breeding soundness exam. He's looking at their body condition score, their feet and leg conformation, examination of the reproductive organs, scrotal circumference, and motility and morphology of the sperm. So I think everybody's pretty good about that, but what most people don't do is a serving capacity test. And to do this, you need to provide a stimulation period for the bulls you're going to test by putting them across the fence from these cows that are restrained for a 10-minute period. And then you always have to have more females in estrus that are restrained than the number of bulls that are being tested. And then you have to make sure the bulls have free access to the heifers or cows that are in estrus. And then you only count the services that include a complete intermission and ejaculation. So most of you are going, man, that sounds like a lot of work. That's a big headache. Is it really that important? If we look at the serving capacity of high bulls or low bulls and look at the percent of cows they detect in estrus, this is at 1 to 100. Those high serving capacity bulls are getting 97%. If you think about that previous slide, we're only finding about 50%. So by having a high serving capacity bull, we're detecting a lot more cows in estrus. Down on these low, it's highly variable, but you're only looking at about 55%. What about the relationship between a bull's serving capacity and herd fertility? These low serving capacity bulls are only finding about 20% of the cows or have a 20% first service conception rate and about a 30% overall pregnancy rate. A medium service capacity bull, about 60% first service conception rate, and that is actually the national average. When people talk about AI doesn't work for me, it's too expensive because I'm only getting a 60% first service conception rate with AI, you're doing just as well as the bull is. Those bulls are 90% overall pregnancy rate, and you go to the high category, you're in the 70% first service conception rate, and nearly 100% of them are getting the cows are bred. We all know to look for testicular size when we're selecting bulls, but sometimes we tend to forget all the things that testicular size can affect. And there is a strong relationship between testicle size and the number of sperm that bull will produce. The testicle size is related to the age of maturity of his daughters. It's highly inheritable. As scrotal size increases, the probability of quality semen increases. So you not only get more semen, the quality of that semen increases. And both buyers and breeders can avoid financial loss associated with bulls that are low fertility by selecting for testicular size. There's a lot of information on this table, but it's looking at testicular size change in Angus bulls with increasing age. And basically at 2 years of age, a bull will have his mature testicular size. There's not a lot of change from 2 years of age down to 9 or 10 years of age, and you do see a slight bump in the testicular size at 9 and 10 years of age, but after 10 years of age, testicular size decreases rapidly. Can you select bulls for fertility? No. It's really lowly heritable trait. However, by crossbreeding, we can increase fertility due to heterosis. If we look at mating performance of bulls as affected by age, the bull ages in this study were either 1, 2, or 3 or more years in age. These young bulls are real go-getters. They have a lot of mounts. But you come down here and look at how many they got pregnant divided by the number they got service, so it's only 40% versus these older bulls are running in the 62% range. Same holds true for the total pregnancy rate. As the bull's age increase, the chances that he's going to successfully have a high pregnancy rate or service cows increases. If we look at abnormalities that affect bulls, and their servicing capacity broken out by what what group they fall into, 82% of bulls were found to be normal, had no abnormalities, and 53% of them had high servicing capacity. But you have to think about 82% were normal, so 18% of these bulls that were just randomly looked at had some kind of abnormality. Bulls with a deviated penis accounted for 5%, and 71% of them had a low servicing capacity. 2% had a broken healed penis. All of them had low servicing capacity. Arthritis was 9%. 45% of them had low servicing capacity, and 2% had overgrown dew claws, and that accounted for 63% in the low servicing capacity. So you might argue, I don't keep bulls that have a lot of age on them. I tend to keep bulls for a shorter period of time, and they're younger bulls, and I'm probably not going to have that problem. This looks at the incidence abnormalities in just young bulls. We're right at that 18%, just like on the previous graph, that have some type of abnormality. 13% had low servicing capacity, 5% smaller soft testicles, less than 1% had penis damage. If we look at the older bulls that are 3 to 11 years of age, 9% low servicing capacity, 9% arthritis, 7% had a broken or damaged penis or a deviation, 2% had overgrown dew claws, and 3% had smaller soft testicles. So the percentage of abnormalities jumped up in this older age group. There are several reproductive diseases can affect fertility of bulls. Trichomoniasis, a protozoan infection, can cause infertility and abortion. Vibriosis, a bacterial infection, causes infertility and abortion. Leptospirosis can also cause abortions, as well as brucellosis, as we all know. IBR is viral, and it can cause abortions and infections in males. And BVD also causes abortions and repeat breeding. So that just emphasizes the importance of not forgetting about our bulls too much when we kick them out for their 10-month vacation. And we have to vaccinate them just like we do our cow herd. There are some other seminal components in the semen that appear to have some effect on fertility. And I'll go through each of these on the following slides. Lipocalin type D synthase is secreted from the epididymal cells of bulls. And in bulls that have above average fertility, this synthase occurs at 3.5 times greater concentrations than bulls of other fertility. It may play a role in both the development of maturation sperm, and it's associated with the plasma membrane of the ejaculated sperm head. How it affects fertility is not well understood or known at this time. There's another lipocalin type synthase, prostaglandin D synthase, and it's also been observed to be in higher concentrations in bulls that have normal to high fertility compared to bulls of all other ranges of fertility. Cytokines and interleukins modulate immune responses. Why would immune response be important? Sperm cells have antigens on their surface that aren't present in the female reproductive tract. So when semen's deposited in the female reproductive tract, it causes an immune response which can decrease fertility. The level of interleukins in bovine seminal plasma has been correlated to individual sperm motility, but not gross sperm motility. So if you look at a whole sample under the microscope and relate that to interleukin concentration, there's no correlation. But if you look at the sperm cells that are fast swimmers, they're going to tend to have higher interleukin concentrations than the slow swimmers. Relaxin is typically thought of as a hormone of pregnancy. It causes relaxation and softening of the ligaments for parturition, but it's also been detected in the seminal plasma of bulls. And seminal plasma concentrations of relaxin have been correlated with spermatozoa that have the greatest motility. The next is heparin binding proteins. Sperm with high fertility bulls have been reported to have a greater binding affinity for heparin, which indicates they have more heparin binding proteins on their plasma membranes. Heparin has been shown to induce the acrosome reaction of bovine sperm. And bulls that have a high affinity for heparin binding protein and sperm membranes have higher fertility than bulls with other heparin binding protein profiles. When we think of prostaglandins, we typically think of female reproduction. Prostaglandin causes regression of the corpus luteum on the ovary, and it was the first compound available for estrous synchronization. So we really think about female reproduction, but prostaglandins were first identified in seminal plasma. And prostaglandin F2 alpha occurs in greater concentrations in seminal plasma than anywhere else in mammalian bodies. However, why it occurs in such high concentrations in seminal plasma has not really been identified. The presence of prostaglandin E and F have been identified in bull semen, and pooled bull semen was found to contain 1,170 nanograms per ml prostaglandin of the F series. This includes other prostaglandins beside prostaglandin F2 alpha. It was also found that extended bovine semen contained 212 picograms per ml of prostaglandin F2 alpha and 311 picograms per ml prostaglandin E2. Well, what's that mean? Does it have any effect on fertility? So what I tried to do was relate those prostaglandin concentrations to a relative fertility rating. Bulls that had known non-return rates were assigned either a low, medium, or high relative fertility rating, these being the most fertile bulls with the highest non-return rate, and the ratio of prostaglandin E2 to prostaglandin F2 alpha was correlated to these relative fertility ratings. And we see as the ratio of those two prostaglandins increased, the relative fertility rating tended to increase. However, the correlation was pretty low. A perfect correlation would be 1. So it's not very good, but it appears that there is something there, and it's very difficult to find very many bulls that have a large amount of data available for non-return rates. When prostaglandin is administered subcutaneously or added to semen before artificial insemination, it results in increased numbers of sperm in the upper segments of the reproductive tract where fertilization occurs in rabbits, ewes, and in sows. When prostaglandin was added to ram or boar semen before artificial insemination, it resulted in improved fertility of females that were inseminated with this semen. However, when prostaglandin F2 alpha was added directly to bovine semen during the extension process, it just wiped out the sperm cells. There was very little post-op motility. But when you do the calculations on how much prostaglandin was added during the extension process, the concentration in the extended semen was about 300,000 times higher than what's naturally occurring in whole semen. So in a subsequent study, I looked at whether or not you could add 0, 500, or 5,000 picograms per ml prostaglandin F2 alpha during the extension process, this being about 20 times higher than normal levels, or 2 times higher, and this being 20 times higher. And this is the normal amount of normal sperm after thawing, and there was no significant difference between the different prostaglandin concentrations. Looking at the percent post-thaw motility of these same treatments, again, they're having a hard time with buttons because I'm having to multitask. You know, I need a cow to help me apparently. But again, there was no significant difference between the concentrations, the post-thaw motility among the prostaglandin concentration treatments. This semen was then used to artificially inseminate cows that had been estrous synchronized, and there was no significant difference between the treatments for first service conception rate. It did appear that there was a slight bump in first service conception rate, but that difference was not statistically significant. And so this tells me that we haven't found the amount of prostaglandin that could be added to semen during the extension process to get a positive response in terms of first service conception and not cause a decrease in post-thaw motility. When prostaglandin F2 alpha has been injected at the time of artificial insemination, it's caused an increased number of embryos in rabbits, improved farrowing rates of sows, and improved conception rate of cattle. I followed up on that study by administering prostaglandin F2 alpha to dairy cows immediately after they were artificially inseminated. And we got about twice as many of the treated cows to conceive compared to the control cows. Should you run out and inject every cow when you AIR? I would say probably not. This is a fairly small study. It's using dairy cows versus beef cows. And I don't know what effect synchronization treatment is going to have on the response to prostaglandin F2 alpha treatment at the time of artificial insemination. So, if you were looking for a magic bullet today, I ain't got it. I don't have anything earth-shattering. And until we better understand the role all those different seminal plasma components play in fertilization or fertility of the bull, and have better methods and easier methods to measure them, I think we have to go back to the basics. And those are providing bedding for protection during cold weather, getting those bulls' testicles up off the ground and preventing frostbite and damage of the developing sperm cells, provide adequate nutrition and vitamin and mineral supplementation, especially thinking about it takes 60 days for that sperm cell to develop. Also, by providing adequate nutrition and vitamin and mineral supplementation, we can try to optimize all those different seminal plasma components that I just discussed. Select bulls with above average testicle size because we're going to increase the quantity and hopefully the quality of semen that bull's going to produce. Get the bulls examined yearly with a breeding soundness exam, which I think the majority of us do. If you have those bulls that you question whether or not they're getting cows bred, Dan talked about maybe doing genetic markers as a way to identify which bulls are not getting cows bred, but then you can also use a servicing capacity test. And then keep our bulls vaccinated against disease, and by doing all these things, hopefully we can maximize the bulls' fertility and keep them king of the hill and get our cows bred.
Are there any questions for John? Or
how many services in your 20-minute time would
It's a 40-minute interval after the 10-minute pre-stimulation, and 5 or 6 services with intermission and ejaculation is a high. On that slide, in that study, they were looking at a 1 to 25 bull to cow ratio, and so that was overall pregnancy rate. On the bottom line of that table was a 30% conception rate for those bulls, or a overall pregnancy rate. Answer your question? Seems low, doesn't it? Right. The national average for first service conception rate among bulls is 60%, but that's just the number bred divided by the number that conceived. But when you look at overall national average for individual bulls, it drops clear down to 40. You've got some bulls that are high performers, and they're probably our high servicing capacity bulls. And then we've got our bad performers that aren't getting their job done.